So now that we have talked about synaptic transmission, we can talk about neurotransmitters and what are called neurotransmitter systems in a little more detail. So what do we mean by a neurotransmitter system, first of all? That means not only the neurotransmitter, but all of the components that go into making and storing and responding to a neurotransmitter that's released from a presynaptic terminal. So it includes, on the presynaptic side, the synthesizing enzymes. So that means whatever enzymes are necessary to specifically make that neurotransmitter in the presynaptic neuron. Now, for peptide neurotransmitters, there is no synthesizing enzyme, or the synthesis happens in the cell body. There may be cleavage enzymes that cut the precursors up, but usually what we mean in this case are enzymes for the smaller molecule neurotransmitters. And then there are the synaptic vesicle transporters. These are the specific proteins that are embedded in the membrane of the vesicle, and they take the neurotransmitter out of the cytosol and transport them into the interior of the synaptic vesicle. So they're the ones that sort of package the neurotransmitter into the vesicles. Again, these are usually specific for the neurotransmitter. So each neurotransmitter has a particular synaptic vesicle transporter or vesicular transporter associated with it. And then the release part, the releasing the neurotransmitters, those components are not considered part of the neurotransmitter system. Here we're talking about the parts of the neurotransmitter, or the step where the neurotransmitter is actually released, the vesicle fuses with the membrane. Those components are not specific to the neurotransmitter. So every neurotransmitter gets released basically the same way. So when we talk about the neurotransmitter system, we're not interested in those, but we are interested in the things that are unique or specific to the neurotransmitter. So that includes the reuptake transporters. So these are the proteins in the membrane of the presynaptic terminal itself that take the neurotransmitter out of the synaptic cleft and move it into the cytosol. Now, synaptic vesicle transporters and reuptake transporters have very similar properties. They both move the neurotransmitter across the membrane, but with the vesicular transporters, they move them out of the cytosol and into the synaptic vesicle, and then when the reuptake transporters move them out of the extracellular fluid into the cytosol. So in a way, they actually are backwards. They work opposite directions from each other, because the cytosol of the fluid inside the vesicles is basically the same as the extracellular fluid. And the reason for that is because of the way that the synaptic vesicle fuses with the membrane. So the stuff on the inside of the vesicle becomes extracellular fluid. And then when the vesicles are recycled, they are recycled. We talked about this in the last chapter. They're recycled through endocytosis. And so the interior of the vesicle, which again is extracellular fluid, becomes the interior of the vesicle after it's recycled also. So that's what we mean. That's what I say when I mean that. What I mean when I say synaptic vesicle transporters and reuptake transporters basically are the opposite of each other. And then in the case of at least one neurotransmitter, acetylcholine, you also have the degradative enzyme. So you have an enzyme that specifically breaks down the neurotransmitter. And again, because that for acetylcholine at least, that's unique to that neurotransmitter, it's considered part of the neurotransmitter system. So those are all the components that are on the presynaptic terminal. And then on the postsynaptic side, you have the receptors that are specific for that neurotransmitter. So again, every neurotransmitter has to have a specific neurotransmitter receptor, a protein that specifically binds to that molecule on the postsynaptic cell. So we talked about those in the last chapter too. Receptors for neurotransmitters can either be transmitter-gated ion channels, which just directly open an ion channel and let ions through that change the membrane potential, or you can have the so-called metabotropic receptors, or sometimes just called the G-protein coupled receptors, that work without directly opening an ion channel, but instead activating one or more G proteins. So the G proteins themselves are also part of the neurotransmitter system, because even though there are each G protein coupled receptor is associated with a particular set of G proteins, and different G proteins can have different effects. And then there are, for some G protein coupled receptors, there are G protein gated ion channels. Actually, this arrow is pointing the wrong way because the idea is that the G protein is the thing that activates the ion channel in this case. And so it's just an indirect way for the receptor to change the membrane potential. But then other G proteins work through second messenger systems. These are molecules that are produced by an enzyme that produces them, and they then go on and activate other downstream molecular pathways in the postsynaptic cell. So these are all the components that are part of a single neurotransmitter system. So for each neurotransmitter, there's a whole suite of proteins and molecules that are associated with just communication, synaptic communication for that neurotransmitter. So if you're going to study a neurotransmitter system, that means identifying those components and finding them, finding where they are. In fact, before you even study a neurotransmitter system, you have to determine if the molecule you're interested in even really is a neurotransmitter, because not all molecules inside a neuron are neurotransmitters, even though, of course, every neuron, like every cell, has lots and lots of different kinds of molecules inside it. So how do you know if a given molecule really is a neurotransmitter and not just a molecule that happens to be found or associated with neurons? So there are a few criteria that we use to decide whether or not a molecule really is a neurotransmitter or not. One criterion is that for a molecule to be a neurotransmitter, it has to be synthesized and stored in a neuron. So in other words, it has to specifically be made in the neuron and then stored in synaptic vesicles. Again, neurons have lots of molecules in them. Many of them might be synthesized by other cells and just taken up through the extracellular fluid, or they might be found in the neuron, but they don't, and made in the neuron, but maybe they don't actually get packaged into vesicles, in which case we wouldn't classify them, strictly speaking, as neurotransmitters. So how do you know if a given neurotransmitter or any molecule is actually found inside a neuron, if it's synthesized there, if it's stored there? So there's different ways to do that. You can look directly for the neurotransmitter itself, or you can look for those other components of the neurotransmitter system. So again, if we back up, if you remember, the neurotransmitter itself should be in the presynaptic neuron if it is a neurotransmitter, but also the synthesizing enzymes should be there. So if you want to know if a molecule is synthesized in a neuron, you can look for the enzyme that converts it from a precursor into the neurotransmitter. So these enzymes are proteins, and you can look for those proteins because they should only be present, or should be predominantly present, inside a neuron if it's a neurotransmitter. Also, you can look for the vesicular transporters, because again, those should be specific to the neuron, and the only job of these proteins is to package the neurotransmitter into the vesicles. So if they're present in the neuron, that means that molecule gets packaged into a vesicle. You can also look for the reuptake transporters, or if there happens to be degradative enzymes specific for that neurotransmitter, you can look for those too. Again, you can look for any molecule if you want to, and there's different ways to do that. Here, what we're showing is a process called immunocytochemistry. So the word immuno has to do with the immune system. Cyto means cell, and then chemistry just means using a chemical reaction to identify something. So the reason it's called immunocytochemistry is because you're using antibodies to try to find these molecules. Antibodies, you may know, are proteins that are produced by your immune system, special cells in the immune system called B cells, and their job is to bind to foreign molecules and things like viruses and bacteria and other infections in your body. And then they act as little markers for your other immune cells, such as macrophages, to destroy them. So every antibody is specific to a structure called an antigen. And the antigen, again, normally would be part of a virus or part of a bacterial protein, or again, just something from outside the body. But what you can do is you can sort of semi-artificially make these antibodies against any molecule you want. In fact, you don't really have to make them at all. You can use an animal host to make the antibodies for you. So if you want to do this, the first step is to isolate the molecule that you want to study. So this is one limitation. You have to know the molecule you're after, and you have to have enough of it in a pure enough form that you can inject it into a host animal. So this is step one. You take your target candidate, and again, it could be the neurotransmitter itself. It could be a purified form of the enzyme used to make the neurotransmitter. It could be the transporter proteins. You can do this with receptor proteins. You can do this with hypothetically with any molecule at all. And then you inject it into a host animal. Now it can be pretty much any mammal. Rabbits are commonly used for this, but you can get antibodies like this from mice or rats, or goats and horses are often used too, and then other vertebrates too. So chickens also, some birds are also used for this. You inject the target molecule into the animal, and the animal should make antibodies to it. Now the catch here is you have to use you can't use a molecule that would normally be found in that animal's body. So for example, if you're going to study a protein in, let's say, a rat brain, you can't try to make antibodies against that protein from another rat, because if you inject a rat protein into a rat, the rat immune system won't recognize it as foreign, and it won't make antibodies. So that's why if you want to study, say, a rat protein, or a human protein for that matter, you would inject it into an animal like a rabbit or a horse, and that would that host animal's immune system would recognize it as foreign. It would create antibodies which would specifically bind to that protein. So then you draw the next step is to draw those antibodies out just by taking blood and then spinning out the plasma liquid part of the blood. That'll contain all the antibodies in that animal's immune system, and then you usually, there may be some purification that you have to do, but then you take a piece of tissue from a brain or from any part of the body. Usually you slice it very thin, or what you can do is grow cells in a dish and apply the antibodies to those. But if you're doing, in fact, technically, if it's cells in a dish, we call it immunocytochemistry. If it's slices of tissue, slices of brain tissue, for example, on a microscope slide, we call that immunohistochemistry. Histo just means tissue. But in any case, you apply your antibody to your tissue, and in so doing, it should bind to the protein or whatever molecule you made antibodies to in the host, in the target cells. But of course, we skipped a step here, because if you just wash your cells or your tissue in antibody, you haven't done much, because antibodies themselves are invisible. So you have to have a way to identify or detect the antibody on the tissue. So really after you draw out the antibody, the next step is to attach a visible marker to it. And so the marker can be just about anything that you can see. So sometimes there are markers that are fluorescent tags, like fluorescent molecules that glow under certain wavelengths of light, or they could be enzymes that you covalently attach to the protein, to the antibody, and that then can catalyze a reaction that produces some sort of pigment or precipitate that you can see, again, under the microscope. But the point is that you have to be able to visualize it. Now, actually, what often happens in immunohistochemistry or cytochemistry is you don't actually tag the protein against your target molecule. So this antibody, in fact, that comes out of the host, we call the primary antibody. And so primary antibody, meaning that it is directly specific for the protein you're after. What's often done, instead of tagging or putting a marker on the primary antibody, is instead you make what's called a secondary antibody. And the secondary antibody is just another antibody that is specific to or binds specifically to the primary antibody. And again, it has to be cross-species. So if you're going to make your primary antibody in a rabbit, then you would make your secondary antibody in another species, like, again, let's say a goat. So you would take rabbit antibodies, not the primary antibodies, just another sample of rabbit antibodies. You don't actually care what they bind to, as long as they have they're made of rabbit antibody protein. Inject that into a goat's bloodstream, and then that goat will make antibodies that are specific to rabbit antibodies, and then that antibody gets tagged. So in other words, you don't necessarily have to tag the primary antibody. You can then tag the secondary antibody, which is sometimes better. It's more specific, and it's usually easier. The primary antibodies, especially if you're talking about a protein or a neurotransmitter that's hard to isolate and hard to purify, then it's going to be hard to make antibodies to, and so the process of tagging them is expensive and time consuming. So the secondary antibody is just a way to do this more efficiently and allow you to do more specific labeling this way. So in any case, you apply the primary antibody. If your molecule is present in a neuron, the antibody will stick. And then you apply the secondary antibody, which again has some sort of visible marker, and attach that to, or wash that over your cells, and that will again make those cells visible. So if you have cells in a piece of tissue, some of which have your molecule, like this one, these little green squares are our neurotransmitter candidate or our protein or whatever we're trying to find, but other neurons nearby don't. So this neuron over here does not have the neurotransmitter we're after, so the antibody doesn't stick to it. And so just those neurons that have your candidate molecule will be present. And this is over here is what it looks like. So this is a neuron that was stained for some sort of protein or neurotransmitter. I don't know what one. And you can see its cell body. You can see the nucleus. You can see many of the neurites. And so that means that neuron has that molecule in it. Now you notice around there's a few other processes, maybe another cell over here that's also tagged, but it's mostly looks like empty space around that neuron. Keep in mind that that's not what brain tissue actually looks like. If you could see every neuron, if every neuron were labeled by this antibody, you would just see a big sea of purple. There would be no real easy way to discriminate one neuron from another. But the purple one just is the one that happens to be positive for that molecule. There are other cells around here. They're just invisible because the antibody didn't stain them. And there's lots of tricks you can do with this. So down here, actually, this is a picture that actually I took back in my my research days. So this is a piece of rat brain that has been stained, actually mouse brain, I'm sorry, that has been immunolabeled for two different proteins, one of which has a green fluorescent tag on it, one of which has a red fluorescent tag. And so what you see is some cells only label with the green tag, which means they have the protein in them that was specific for one antibody. And then the red cells, the ones that are actually yellow, have both the green and the red. So this is the way fluorescence works. If you have two fluorescent tags and then kind of overlay them in a computer image, you'll get in this case yellow. So the yellow cells have two different proteins that I labeled with two different fluorescent tags. And there's lots of other interesting tricks you can do with antibody interactions like this. And so this immunohistochemistry or cytochemistry is a really good way to, again, find particular molecules within a piece of tissue or within a cell. Sometimes you can even use it to find within the cell itself. So some proteins may be expressed throughout the cell. Some proteins may only be found, let's say, in the axon or in the dendrites or at the presynaptic terminal. But in any case, you can directly see them in the cell right here, and so you can find, maybe not only which cells contain them, contain your protein, but where they are in the brain or where they are in other tissues. Another thing you can do, another technique to find which neurons contain a given protein or not, is something called in situ hybridization. So, so in situ means in place. So basically, it means you're doing it in the cell itself, as opposed to in a dish. And in situ hybridization is a way, instead of looking for a protein or another type of molecule with an antibody, you're looking specifically for an RNA molecule, or usually it's RNA, and usually messenger RNA. But you can do this, you can use the same technique to look for any nucleotide sequence. So it could be other kinds of RNA. You could even do this to look for DNA. But you should know that mRNA stands for messenger RNA. This is the RNA sequence that gets made when a gene is transcribed. So a DNA sequence is transcribed into an RNA sequence, and that RNA sequence gets translated into a protein. So for any protein that's found in a cell, that protein also should have an mRNA sequence that's also present in that cell, because otherwise, if not, then the protein itself wouldn't exist. Proteins can't really jump from one cell to another, so any protein you find in a cell must have been made in that cell, and that means there must have at some point been an mRNA specific for that protein. So this is another way to look for proteins. Instead of looking directly for the protein, you look for the mRNA that codes for that protein. And it uses the fact that mRNA is a single-stranded sequence of nucleotides, and you can make, if you know the sequence, you can make another sequence that's complementary to it. So if you know what that means that let's say if this is the mRNA, I don't know what these different shapes are, but let's just say this is A. We'll say this is C, and maybe that's G. And then this would be T, and then this would be another C, and so on. So if this is the sequence of the mRNA, what you can do, if you know that, you can make what's called a probe. This is another sequence that is complementary to the first one. So if there's an A in our mRNA that we're trying to find, then there needs to be a T here. If there's a C here, there needs to be a G across from that. And if there's a G over there, there needs to be a C here, and so on. And so this whole probe then is complementary to at least part of your mRNA. It doesn't have to be complementary to the whole thing, but the more of it it's complementary to, the more specific it will be. And then you put that on your cells or on your tissue, and again, if the mRNA is present, then the probe should stick to it, because that's how nucleic acids work. If you have two complementary strands of nucleic acids, they will bind to each other. Now again, our nucleotide, these probes are invisible, just like antibodies are by themselves. So you need some way to make them visible, some way you can actually see them inside the cell. In the old days, they would make these radioactive, which meant that when they made them, they usually use some sort of radioactive isotope. So oftentimes it'd be radioactive carbon or radioactive hydrogen. And sometimes that's still done. It's done less nowadays because radioactivity is just expensive and really difficult to work with. So now there are kind of safer and easier probes that are using, again, fluorescent tags or maybe enzymes or just other substances that are easier to work with. But in any case, you apply that to your cells or to your tissue. If your gene is being expressed in that cell, which means it's being eventually a protein's being made from it, then the probe will be present. The probe will stick to the mRNA, and it will make that cell visible. Meanwhile, adjacent cells that don't have your RNA in them will not be visible because the probe won't stick to them. And then you get something that looks like this, where all these little purple dots here are neurons, or cells in any case, we don't necessarily know if they're neurons, that have that mRNA, which means they express that gene. So this is just another way to look for proteins. Now, of course, it's only good to look at for looking at proteins. With immunohistochemistry, hypothetically, you can look for any kind of molecule. So you can look directly for neurotransmitter molecules, or again, anything that you can make an antibody against. Whereas in situ hybridization only works on mRNA. So that means that you're only looking at those proteins. So you can do this for the mRNA sequence that's associated with, say, the synthesizing enzyme, or for the transporter proteins, or whatever component of the neurotransmitter system you want. And again, you can do this for any kind of gene at all. It doesn't even have to be associated with neurons. You can do this in any tissue. You can even do this for sequences that aren't even going to be made into a gene. So it's a really useful way for just finding which cells have these genes turned on. The only catch with in situ hybridization, again, you can only look for genes, and as we know, the mRNA for any protein in a neuron will only be found in the cell body and sometimes in the dendrites. So in other words, you can only find the cell body. You can't, let's say, find the exon associated with that protein. So if we look up here, this purple cell, we can see there are processes coming out. Now I don't actually know, these look like dendrites, but there could be, one of these could be an axon. And in fact, in principle, what you could do is look in a different part of the brain, and if the protein is found in the axon or at the axon terminal, you can even trace the axon back to the cell body. You could figure out where the axon goes and actually map connections that way. But you can't do that with in situ hybridization because it only tells you where the cell body is. It does not tell you anything about what's going on necessarily at the axon terminal. But either one of these techniques, and there are other ways to do it as well, will tell you your answer to that first question. So is my neurotransmitter or my thing that I think is a neurotransmitter actually found inside of neurons? Now, again, this you can look for the neurotransmitter itself, like I said, or you can look for the enzymes, or you can look for the specific vesicular transporter. So that'll answer the question of not only is it synthesized, but is it stored in those cells? And you can look for the reuptake transporters too, and so on. So any component of the neurotransmitter system, you can use this technique to find. And sometimes, in fact, you need to do all three. Now, the next question, or the next criterion for a neurotransmitter, is that it gets released from the presynaptic terminal when the axon, or when the neuron, is stimulated. So that means if the axon experiences an action potential, then the neuron must release that neurotransmitter from its terminal. Now this criteria is harder to determine because when a neuron, the only way to do it is to directly stimulate a neuron with an electrical current or an electrical voltage, and then collect the fluid from around the place where the synaptic terminal is. This, in fact, is what pretty much what Otto Loewi did when he identified the fact that the neuron, the nerves that innervate the heart, the vagus nerve, releases a substance that causes the heart rate to slow down. He took the heart out of a frog with the vagus nerve attached, and then electrically stimulated the nerve and drew fluid from around the heart and applied it to a different heart. And so what he showed is that the substance being released from the nerve is the thing that caused the heart to respond. And again, he could do this because this was possible with this is easier to do with peripheral nervous system synapses, like at the heart or along any muscle, because you can isolate the synapse in, and usually remember that the neuromuscular junction, this is an example of, is very reliable, which means that the neuron releases a large amount of neurotransmitter. And so if you incubate it in fluid, you can draw that fluid out and you can look for the presence of the neurotransmitter. And it doesn't even have to be, again, from heart tissue. You can do other ways to identify the neurotransmitter in your sample. But in the brain, this is harder to do because it's very hard. Neurons are small. They only release a tiny, tiny amount of neurotransmitter. And so it's hard just mechanically to isolate the fluid from a single synapse. And especially to isolate it from other molecules that may be present in the extracellular fluid. So a lot of times this is done with large numbers of neurons or pieces of brain slice that are kept in a dish and done with many, many samples over and over again to collect a large amount. But in any case, all you need to do is show that when you electrically stimulate a neuron, your candidate neurotransmitter is being released. And then the last step, or the last question to answer is, does your candidate neurotransmitter produce a response in a neuron that matches the response when you electrically stimulate a presynaptic terminal? So again, this one's kind of complicated. You have to first identify a neuron that you think releases your neurotransmitter. So that's this neuron, that's this axon here. So your candidate neurotransmitter has to be, or you suspect is being stored and synthesized in that presynaptic cell. And then you also have to find a postsynaptic neuron, a neuron that is connected to the presynaptic cell. And then when you electrically stimulate the presynaptic neuron, it has to give some sort of specific response. So it doesn't matter what the response is, but let's say here we've stimulated this axon, and then down here, this is the response we get, this train of action potentials at this particular rate. And we record that with a electrode in the postsynaptic cell. Now, though, what we have to do is say, okay, does our candidate neurotransmitter do the same thing? And to figure that out, you again usually do this in a culture dish or maybe in a brain slice that's being kept alive in a dish, and you apply a small amount of your candidate neurotransmitter to the neuron. They're using a process called iontophoresis, which instead of squirting the neurotransmitter on using pressure, they use an electrical current, which causes the substance to move out, and then that way it's just a tiny bit. It's not being flooded or washed away by your neurotransmitter. And if when you do that, you get the same response. So if applying the neurotransmitter or whatever substance you think is a neurotransmitter to your cell does the same thing as electrically stimulating the presynaptic terminal, then that's your clue that in fact your candidate molecule is a neurotransmitter that is released from the presynaptic that presynaptic axon. So again, those are the three major things you have to do to show that a neurotransmitter or a molecule you think is a neurotransmitter really is. Because again, there are going to be other molecules in a neuron, some of which are neurotransmitters, some of which aren't. And so to show that it really does work like this, you have to really do a lot of work.